I'm Dr. Kirsten Traynor, presenting on superorganism resilience of Apis mellifera colonies. The idea of resilience is that the superorganism has a built-in ability to preserve the germline and only sacrifice some of the somatic cells when faced with stressors. That is, a honeybee colony with up to 50,000 individuals can afford to lose some of its reserve workforce and still survive. I investigated how exposure to sublethal pesticides and pollen impacted this resilience. Honeybee colonies are highly eusocial, with the division of labor, cooperative brood care, and overlapping generations. They are considered a superorganism, a group of synergistically interacting organisms with one reproductive queen, who functions as the germline, and thousands of typically non-reproductive workers that function much like the somatic cells in a human body. Honeybee workers forage in their surrounding environment for carbohydrate-rich nectar and protein-rich pollen, which they bring back to the colony. The nectar is used to fuel the activity of the adult workers or turned into honey, while the pollen is stored in the colony where it is consumed by nurse bees that turn it into protein-rich brood food. The queen lays eggs, which hatch into larvae after 3 days. These are then fed copious amounts of brood food by the nurse bees for 6 days, whereupon the cells are capped. Worker bees emerge as adults 21 days post-egg laying. We ran 2 large field trials. One in 2018 involved 24 full-size colonies, and a follow-up experiment in 2019 involved 96 mini-colonies. For each trial, we had 4 treatment groups. The bees received pesticides mixed into a pollen patty. The insecticide group was treated with chloripyrifos and fenpropathrin. These were the 2 most commonly detected insecticides in a study of commercial colonies. We incorporated them into the pollen patties at 10% of a honeybee's LD50 if she consumed it over her entire 10-day nursing phase. The fungicide group was treated with chlorothalonil and propiconazole, the 2 most commonly found fungicides while bees were pollinating blueberries. As fungicides are considered bee safe, we incorporated them at a rate of 2% of a honeybee's LD50. The both treatment group had both the insecticides and the fungicides mixed in. Colonies were fitted with pollen traps to remove any incoming pollen from foragers and then fed the contaminated pollen for 4 weeks. In the large-scale field trial, we found that the different pesticide treatments had a significant effect on pollen consumption. The fungicide group consumed less pollen than controls, while the both group consumed less than all other groups. We measured honeybee foraging over 5-minute intervals for each colony on multiple days, noting if the forager returned with pollen or was a non-pollen forager. Non-pollen foragers could be carrying nectar, water, or nothing. The control colonies had significantly different proportions of pollen to non-pollen foragers compared with the other treatment groups, dedicating more effort to non-pollen foraging. We caged the queen and then measured brood loss of 50 eggs during development. The insecticide group had significantly higher brood loss during the larval stage and total loss throughout the experiment compared to the control group. We also monitored queen turnover events in the colonies for the month we fed the contaminated pollen and for the 3 subsequent months. The fungicide and the both group experienced significantly more queen events over the season than the control group. We repeated the experiment in 96 mini-colonies to better observe queen events. All 3 treatment groups had significantly more queen events than control colonies. In miniature colonies, the bees will sometimes abscond when they get overheated, abandoning the hive. This differs from swarming in that they do not draw out a queen cell prior to departure. Significantly more colonies in the fungicide treatment group engaged in swarming behavior than controls. In summary, sublethal pesticide exposure has wide-reaching impacts on the honeybee superorganism, leading to reduced brood viability and increased queen events. We examined only 4 commonly found pesticides in pollen, but as this network diagram demonstrates, there are many different combinations detected together in pollen, allowing for a large variety of potentially synergistic interactions. I would like to acknowledge the diverse funding sources that made this research possible, as well as individuals who assisted in this research.